Yo, yo, yo. Welcome to Enzymes. Okay, so in order to understand enzymes, we need to take a look at what a chemical reaction is because enzymes are biological catalysts and the catalyst speeds up a chemical reaction. So, with chemical reactions, we have our reactants. And the reactants, again, break bonds and then form new bonds to rearrange to become products. And with that said, we have bonds being broken, bonds forming, and that can only happen if there's enough kinetic energy when these molecules collide. And when the molecules collide, they also need to collide in proper orientation for the products you see here to form. Okay, so we need to know the different parts of the enzyme. I'm going to rewrite this part for y'all. The enzyme, again, is a biological catalyst. So that catalyst is not part of the reaction itself. It simply speeds up the reaction. The first part you need to know about enzymes is that the reactants are actually called substrates. So these two terms are interchangeable. And the reactants, again, bind to the enzyme itself at a place called the active site. So the active site actually changes shape to wrap tightly around the substrate to form this complex here called the enzyme substrate complex. Once bound, the substrate is changed into the products that we see here. So those are the three important parts: the substrate, the active site, and then the enzyme substrate complex, which changes the substrate to the products. There are three properties you need to know about enzymes. The first one, again, is that it lowers the activation energy of a reaction. So the activation energy can be seen here, highlighted in purple. Okay, is from the reactant's free energy to the very peak of this curve. Now, notice that the enzyme lowers the activation energy alone. The free energy of the products and of the reactants remains the same. So the first property, I'm going to write down here, lowers the activation energy of the reaction, but does not lower the free energy of reactants and products. Then we have our second property. The second property is that the enzyme can be used over and over again. So used over and over again. It isn't altered by the chemical reaction itself. It will remain the same even after the products are produced. So again, it cannot be used up. It can be used over and over again. The third one is that the uh, substrate has a very specific spot, and so that enzymes are also specific. They catalyze one reaction. It doesn't mean that only one substrate can bind there. Other molecules can occupy the same active site, forming that weak interaction, blocking the substrate from binding to it. But if it is not the substrate, it is not bound, and it cannot be uh, broken apart. It cannot be changed into the products. So again, if a molecule can bind to that active site, it doesn't necessarily mean that that would also undergo the same reaction that the substrate does. We are going to dive into the regulation of these enzymes. So the first type of regulation that increases the rate of reaction, as seen here, is to increase the substrate concentration. By increasing the substrate concentration, more enzymes will bind to the substrates, as seen here, and so there will be a higher rate of that reaction. But increasing the concentration can only go so far, up until this point here. At this point, we have so many substrates, but all of the enzyme's active site are bound. So it is working at the fastest rate possible. In order to bypass this reaction rate, to make it go even higher, as I'm highlighting, all you need to do is to add more enzymes. By adding more enzymes, we have more active sites for these substrates to bind, thus increasing that reaction rate to break down those substrates into products. Enzymes work in optimal conditions. So this active site is very functional and efficient at certain optimal conditions. And so again, enzymes work in optimal conditions, usually described as temperature or pH. So outside this range of tolerance, one would say, the enzyme would denature, so the substrate would not turn into products after a certain range. So let's take this temperature example. So this enzyme is at the optimal temperature for typical human enzymes at 37 degrees. So at 40 degrees, it is still, you know, efficient, but not as efficient as that 37 at this peak here. So the enzyme starts to denature when it reaches zero. And it looks like it's about 43, 44 degrees Celsius. So at this rate, or at that temperature, the enzyme is considered to be denatured, and the opposite side is true as well. At this here, 0 degrees Celsius, the enzyme has denatured too. So again, denatured doesn't mean just being outside is optimal. There are ranges outside the optimal, as I'm highlighting right now, that is still considered to be very efficient. But if we move further or further away, it gets less and less efficient, and again, becoming dangerously to that denatured state of the enzyme, where it no longer will break down that chemical reaction. So we know that DNA leads to RNA, specifically mRNA, and the RNA is translated into proteins, and those are the enzymes we're talking about. Well, mutations occur at this stage here. Thus, if there's a change in the DNA, then there could be a change in the RNA, thus a different amino acid, thus a different folding to the enzyme that we are looking at. And so this is the example illustrated. We had a hydrophilic amino acid, and it got mutated into a hydrophobic amino acid. Well, this hydrophobic amino acid will cause the enzyme to change its shape to the point where the substrate can no longer bind to that active site. Thus, it cannot um, catalyze that substrate into the product anymore. So mutations again limits the efficiency of the enzyme. We are going to be talking about special classes of enzymes. So the first one requires something called a cofactor. So the cofactor is defined as a non-protein helper. Okay, there are two types. The first one is considered to be inorganic, and the example would be your uh, zinc, your iron. Okay, so those would be considered your inorganic cofactors that help once bound to the protein makes it active, exposing active sites for the substrates to bind to. So again, cofactors are a required part for these enzymes to work appropriately. The second type would be organic cofactors, and organic cofactors is special because they have a special name. They're called coenzymes. They work the same as these cofactors being a requirement, binding to the enzyme, and then once bound, it exposes some active site for the substrates to bind to so that it can change into the products. So again, two types of cofactors, inorganic, and we can simply call those cofactors. Or we have the organic, and they have the special name, coenzymes. I forgot to give you the example. It would be your vitamins. Okay. Okay. Our second special class of enzymes again have something that could block the active site or change the active site. And they are called inhibitors. So these inhibitors bind to either the active site or the enzyme itself. And when it binds to the enzyme itself, but away from the active site, it changes the active site. So let's talk about it. We have the competitive inhibitor. When I see that word competitive, I will think about an inhibitor that competes or fights for the active site. So it can bind the active site. So I'm going to say that it blocks the active site and it prevents the substrate from binding to the active site to get it catalyzed. The second type would be our non-competitive. So this non-competitive inhibitor doesn't fight for the active site, but instead it binds to a different spot on the enzyme and changes the active site so that the substrate no longer matches the active site shape and can no longer bind to it, thus making the enzyme non-functional essentially. There again, are two types of inhibitors on top of the non-competitive and competitive. And these inhibitors, again, are going to be described as how they bind. They can be either reversible or irreversible. For the reversible, these are weak interactions at the active site, like hydrophobic interactions, hydrogen bonding, all those things are considered to be weak interactions that are temporary. But then irreversible inhibitors form a covalent bond with the active site or with the enzyme to change its shape because again these reversible or irreversible could still be competitive or non-competitive. But again, irreversible inhibitors covalently bond to the active site or a part of the enzyme, and then again making the enzyme non-functional. So, when we're talking about these irreversible or reversible competitive inhibitors, irreversible, we really can't overcome that inhibition, but we can overcome the competitive inhibitor, okay, as long as it's reversible. So again, the way to overcome the competitive inhibitor is to increase the substrate concentration. When we increase the concentration of the substrate, there is a higher probability that the enzyme will bind to the substrate rather than the inhibitor. Okay, so again, this is true as we have seen it before. This is the idea. So again, if there's equal amounts of substrates and competitive inhibitors, 50-50 chance that it will bind, as seen here. But if we increase the concentration of the substrate, there's a higher likelihood that it would bind to the active site rather than the inhibitor itself. There's no way to outcompete the non-competitive because once bound to the enzyme, it just makes the enzyme inactive. And this is the highest rate that would occur. Now, we're going to talk about another set of special enzymes, and they're called allosteric regulators. So these are enzymes that can be turned on or off. That's the key thing. They can be turned on or off to increase a certain product or to decrease a certain product. So there are two states to it. There are the activators. In terms of activators, they start off as inactive enzymes where the active site is not appropriately shaped to accept the substrate, but once the regulatory molecule called the activator binds to the allosteric site, the inactive enzymes then becomes active and thus increases the desired product. So again, this allosteric activator is called a regulatory molecule. So another molecule can come in and turn on an enzyme if this was in an inactive state. But there's the opposite. There are inhibitors. So allosteric inhibitors, again, the enzyme starts off as active, catalyzing the substrate to products. But once there's enough, there would be a regulatory molecule that's called the inhibitors, but binds to the different part of the enzyme, thus making it inactive. Now, one would ask, hey, Mr. Tran, what's the difference between an allosteric inhibitor versus a non-competitive inhibitor? Well, I would say that essentially everything and all of the concepts around those two ideas are the same. Some sort of molecule goes in and binds to a different part of the enzyme, shutting it down. But there is a big difference between the word allosteric regulators and non-competitive inhibitors. So with allosteric regulators, they can be turned on or off, whereas the inhibitors would just turn the enzyme off once bound to that other site on the enzyme. Finally, we have reached the end of our notes for enzymes. This here is called a feedback inhibitor. In a feedback inhibitor, the substrate is being converted into very, very specific intermediate substrates or middlemen before reaching the desired product. But in this specific example, the product actually inhibits enzyme 1. So one question that could be asked on the exam is what would happen if the end product interrupts enzyme 1? And we will only talk about the immediate effect because once we have the effect running for a while, again, homeostasis will be reached and everything will reset and it will start over again with more of this substrate being produced. So if the substrate cannot be catalyzed to intermediate substrate A, there is a buildup of this triangle substrate. We could also ask what happens if the end product inhibits enzyme 3. Well, everything would work up until that substrate B is trying to be catalyzed to that end product, and so there's a build up of substrate B. So that's the concept of the feedback inhibitor. The end product will inhibit an enzyme earlier on in the pathway, stopping the production of the product. Okay, y'all, that's it for today. Have a good one.